While we get everything going, there is a new computer in here this year, so if you're used to the old settings, be prepared. You will have to re-log in and all that is. Thanks everybody for showing up this whole semester and enduring the chaos that is life in the United States at this point in time in history. Welcome fall semester. Our first speaker, I'm just going to go super fast, is Nate Lewis. He is going to tell us about his research. He is applying for a courtesy position in IOB, so I hope you'll find his research entertaining, and we'll ask him many questions about how it relates to what you're working on.
Thank you. I want to thank you guys for I just moved my lab this past year, and actually my family just finally made it out here a few weeks ago. So we're settled here mostly, but still living in a tiny apartment in our house. So, but anyways, a little bit about myself. I'm a systems biologist by training and so forth. I think about every living thing as the molecular networks that make it up and how that leads to the phenotype we ultimately see. Not only that, sometimes we can tinker with those networks, right? A lot of therapeutics are basically perturbing not necessarily individual genes, they're perturbing various dysregulated networks. And so if we go in with that mindset, we try to figure out what those networks are, we might be able to find better therapeutics. Or, another area of my lab, we can actually manufacture a lot of drugs using living cells. And so we need to understand how the cells are making those drugs. And then you can perturb those and make better pharmaceuticals. And so today I'm just going to give one little story of some stuff we're doing. One of the little niches of our research, which happens to be in a little area. It started out when I was a new faculty member, someone asked me if I was interested in doing this, and I thought about the data that we're going to be generating, and I'm like, that would be really cool, and it'll be applicable to the stuff I really care about, but I really don't care that much about it. And that was around the autism spectrum. And then lo and behold, all three of my kids got diagnosed. So, okay, I really care about this now. I think it's a really fascinating area because many of us are wired a little bit differently. And if we can understand that wiring a little bit, it opens up opportunities to understand each other better. And in some cases where there might be room for therapeutics and so forth, that we might be able to make better therapeutics to be able to treat some of the negative phenotype or traits, some of the struggles that I've seen in my kids and so forth. And so today I'm going to be talking about how we're thinking about neurobiology and thinking about autism, the autism spectrum in particular, as an alteration in intercellular communication. Okay, and so I'll be able to start.
You might have to reactivate it. Just click it one time there, and then it'll go.
Okay, there we go. Okay, so this is a picture. Fortunately, it didn't mirror today, but one of the last times I gave this talk, I was sitting in my office, and a student runs into my office and said, "Nate, aren't you supposed to be giving a talk right now?" And I was like, "No, it's an hour from now. I still have time." She's like, "No. My friend called me. He's in the class. You're supposed to be there now." I looked at my schedule. Sure enough, I'd gone wrong. One of the problems with scheduling things while you're in other time zones. But fortunately, it's not today. But anything you're doing, whether you're running to give a late lecture or you're sitting there trying not to fall asleep, these interactions, it's the molecular and cellular interactions are defining life. That's really influencing those functions. Your muscles, I mean, you have these interactions between individual cells are telling you how to contract or relax and so forth. And you have right here, each individual cell is not on its own. It's sending out these signals to each other, sometimes protein ligands, sometimes small molecules, metabolites, and there's receptors receiving those. And that's telling the next cell what to do. And now a lot of these signals, I like to point out one area that I'm particularly interested in is in the protein secretion pathway. You have the endoplasmic reticulum and Golgi are basically processing a large percentage of these signals and receptors. And so we need to start thinking about the molecular level inside of the cell and the molecular level outside of the cell. What is the signal being sent out? And so these signals, whatever they be, wherever they're coming from, they're going to influence differentiation, host microbe interactions, immune system, neurodevelopment, basically anything you're studying. There's going to be some ligands and receptors interacting to tell the cells what to do next. Okay. And so this is going to happen through direct cell-cell contact. You're going to have sometimes secreted signals, relining receptors. Sometimes the signals are going to be on the extracellular matrix, waiting for a cell to interact. And so we need to start to understand these molecular processes. And so the idea here is, though, to understand them, we got to figure out how to interpret those signals. What is the hot, juicy gossip that the cells are saying to each other? Okay. And so there's a number of ways. We have decades of amazing research where people are using detailed biochemistry, cell biology, molecular biology, all these different techniques to tease apart these individual signals. Now, we were building up large repositories of ligands and receptors where we know that, for example, this protein is going to be secreted. It's going to land on this receptor, and that's going to then stimulate a signaling pathway and change transcription in the next cell. And so now that we are accumulating these thousands and thousands of known interactions, we can start thinking about using larger data sets to interpret what exactly are the cells saying to each other. So what you can do is you can take a collection of samples or cells. We do a lot of single cell work. And so you can take the transcriptomes of individual cells, and that will tell you which ligands or which receptors are expressed in that individual cell type. And so you take your data set right here, you got your genes by your samples, and you get the expression levels. To simplify things, we will trim these down to the known ligand receptor pairs. So you have a smaller data set, easier to work with. And then what you can do is there's a number of different techniques. I won't go over the details on this because there's different ways you can do this, but what you're trying to do is you're trying to find correlations between ligand expression in one cell and the receptor in the other cell. And if you can start to understand start to get that information, get the correlation, we can start to make assumptions that maybe as a ligand changes expression in one cell and the receptor changes expression in a neighboring cell, that's a prediction in a way that there might be communication going on between those. Now, as we're getting more techniques, such as single cell proteomics and metabolomics and so forth, we can start to think about these signals and start to detect these signals a little bit more physically in your sample of interest. And so, but once you get these, once you identify the ligands and receptors that are correlating well, you get a communication score, and that will then give you a metric that you can go through and do further analysis. You can plot it, make pretty diagrams, and so forth, and then go in and try to experimentally validate and study and perturb those ligand receptor pair interactions. And so that is a way that you can start to clue in on a single cell data set on the hot, juicy gossip going on in that sample at that moment. And so the question is, gossip doesn't always lead to action. And so there's certain communications, certain conversations that lead to action. We want to understand what are the important messages? What are the ones that are going to get the cells to make a decision and go do something else? And so the challenge here is that even if any of you've worked with single cell transcriptomics or any single cell omics, it's a big data set. It's hard to work with. You wrestle with it. You're not sure if you're normalizing things correctly. You're not sure if the UMAP that you're looking at, where you've done clustered all the different cells, whether that's reality or whether that's just an artifact. Okay, it's a lot of work. Now, just to make things even more complicated, is that biology is not a snapshot, a single snapshot. It's not a single sample. It's dynamic. And so we're dealing with things like you're going to have multiple time points of stage. If you think about doing single cell omics on hematopoiesis, as the cells are differentiating and so forth, that's going to be important. That's going to be your signals, and you see the responses, right? You also have different disease states and levels of severity of disease. Something I'm not going to show, but when we first developed this, one of our tools, we had the first data sets that came out of the COVID pandemic. where we had multiple levels of severity of COVID patients. And so with that, you can leverage that information to see, well, maybe the signals that are more, as they get stronger, that they actually might be the most important signals that determine patient response and severity. You also have cellular location, so it's also spatial issues. And so the challenge here is when we started working on this stuff, when we were publishing our that COVID study at that time, especially, the best tools out there would allow you to look at one sample at a time. You could look at the communication of one sample. And if anything, you think about stats, like my brother's an economist, and he looks at me and says, I don't trust anything you guys do because your number of samples is too low. I never told him about the N of 1 in early single cell omics. And so in all reality, we're getting bigger data sets now, and we can get so much more information on this. And so the question is, how do we leverage the dynamics across multiple samples when the best of the tools out there can only handle one sample at a time? There was one actually at the time that was able to handle two. And so what we did here is we were looking at this, and you could get for one specific ligand receptor pair interaction. Let's choose your favorite secreted protein and your favorite receptor. You can look at that one. You can look at, for each individual cell, you can look at the expression of all genes, break it down to that single ligand receptor pair. And there's your UMAP, so you can have the different cell clusters. So you can get the individual cell types right there. You can start to look at correlations between the cells in one cluster, such as that one, versus that one. Do you see correlations of the ligands and receptors in those? And with that, you can then get your communication score to say, well, cell 1 and cell 2 are likely communicating you can say whether or not they are likely communicating with that one ligand receptor pair. So here's all the individual sender cells and all the individual receiver cells. You can look at, is there a correlation between their expression and the different cell types, cell clusters between those. And you can look at all those comparisons there. And that gives you a matrix of communication, of likelihood of communication between cell type 1 and cell type 2. And I mean, that's nice from one sample. Fortunately, we have really great experimental stuff that will help us identify what are the false positives and false negatives when you do the hard experiments afterwards. If you're a bioinformatician, the experimentalist might not like you because of your false positives, but hey, it's part of the game. So what you could do, however, is if you want to go in, you can now repeat this process for each and every ligand receptor pair. And with that, you can start to see you got more matrices, more data to analyze, more things to worry about. We want to go, that starts to give you more information, but again, remember, this is one sample. So you can take this and you can put it in a 3D communication tensor. So in other words, a three-dimensional matrix of all the ligand receptor pairs, and you can look at correlations. You can identify, okay, what are the ligand receptor pairs used in that one sample, which cells are being involved, and so forth. But again, I mentioned biology is dynamic. We don't care so much about one cell in isolation. We care about how it compares to many others, whether it be other patients or other time points in a trajectory. And so that's where we started thinking about, the nice thing about tensors is that you're not stuck to 3 dimensions. We may only be able to really imagine them in 3 dimensions. But there's a lot of great math out there that's been used. Some of the very earliest techniques used for image processing came from singular value decomposition and tensor decomposition, that sort of stuff. And so my student who was developing this, we were at UC San Diego at the time, so we always bragged about, we always had to show off the peer at our university because, hey, we had a peer. I got to get a really good one for UGA now. But basically what you do, you can take a photo, and a photo is basically a 3-dimensional tensor. You have your X, Y, and then RGBs, different color channels, okay? And you can take this and decompose it to find the major features in that. What do you think the major features of that photo might be? When we break it down to the 3 dominant feature spaces you have right here, you can kind of see the pillars pop up. You can see the directionality for the periods and the ambient light. You see kind of the bands of light coming through there. The second factor is going to give you the light between the pillars and the ocean waves. You can see the light reflecting off the waves. And then we get right here in the third factor, the light reflecting off the wet sand. And so you can separate these. And then that basically what you've done there is you have broken it down. Really, these represent just a few vectors. And so it's a way to separate it and decrease the amount of information, or decrease the amount of data you have to transmit if you send. So similar approaches were used in the early moon landing missions to send the photos from the moon back to Earth, because it was a way to easily compress and restore the data, because you could then add these back together on the other end of the decreased information right there and reconstruct it. Now, you definitely would want to go with more than 3 features or 3 of the factors. But this kind of gives you an idea that you can start to reconstruct it with a limited number of factors and so forth. And so tensor decomposition, what it is giving you, it's giving you the major signals that are in this complex picture. So we were thinking we could actually apply this to cell-cell communication, where basically you have right here, here's your complex communication tensor. And within that, you have individual cells, and different layers, different ligands and receptors that are being used between those cells. You see that dimensional space, there's different communication going on between different group cells, represented by these different patterns. Okay, so we can take single cell data and decompose, pull out the individual signals, is what we were thinking. So yeah, so factor 3, you can see 2 cells are communicating using a specific ligands or receptors as represented called yellow. And so this is nice and all. I'm still looking at 1 sample. What about more samples? Again, with tensor decomposition, with tensors, you can do as many dimensions as you want. And so what we could do is now we can start to take every sample and have the samples of itself be a 4th dimension, the tensor. And so with that, we can now lump those together and do the tensor decomposition. And what we'll get there are individual factors. And these factors, you can think of these as different conversations that are happening in your sample. And so what you'll get when you pull it out, because none of us can really imagine something in 4D, maybe some people are smart like that, but what you can do here is you can break it down to these different plots, where right here, those will show you for each individual sample for this factor. Let's say this is factor 2. You would have certain magnitudes of strength of in that factor, where this first sample is going to be really important. So if you think of a time course, it would say that the communication in this one best represents what's going on in the very early stages of development. If you're looking at a sample different, or some cells differentiating, and then the signal is going to be decreased in the second sample, and so forth. And so you get a sense of which samples have this communication going on. And what you have here in that communication, you have certain ligands and receptors that seem to be very strongly infused especially in this sample, a little bit less in this one, and so forth. And then this tells you which cells are going to be involved the most, which ones are going to be highest, whether it be like this one right here is going to be the most dominant one, sending out the signals, and this one right here is going to be receiving. So it looks like it's actually responding to itself, because these would be ordered, these would be the same cell, or the same cell cluster. And so we can now start to do this. Well, this is just one conversation going on in that whole mess of cells. And so what we did, we were wondering if this really was real. And so what my student did, Dr. Bagassarian, he put together a simulation where he simulated 3 cell types, or 3 groups of cells, all communicating with each other. We have some cell type A is talking with itself, but also with cell type C. Cell type C is only talking with cell type A, and cell type B is only talking with itself. And he ran the simulation, and then built like a kind of a fake single cell transcriptomics data set based off of that, and then ran it through our approach. And he had right here these individual signals. Not only were was the communication happening as described in that little diagram there, it happened on a time course representing either a sawtooth wave, where one of the signals was increasing and going down and up and so forth. We had another one where there was a pulse, and another one where it just rapidly dropped off and flattened out. Another one that decreased linearly. Okay. And so with this simulation among these three cell types, he went through and one of the first steps you do is you figure out how many signals are there. So you can do an elbow plot, and with that, he found that the data set had a rank of 4, so there was 4 signals. Good, that's what we were trying to put in there. It's 4 signals. And when he decomposed it, he was able to reconstruct the different ones, representing each spot representing a different time point and a magnitude of the signal, and it gave the ligands receptors. We used a large number of different ligands receptors in here, and it was able to capture all of those, and it accurately told us that cell type A was talking with cell type C. They were communicating with each other. And we also saw that in this signal, that was the one where cell type C was talking to cell type A. And so you can go through and you can start to pull apart who is talking to who, what are they saying, and at what time points, by decomposing that single cell data. Again, this was simulated single cell data. So as I mentioned, we did go through and we did apply it to COVID severity. And it was the funniest part about this was you get a paper written and you submit it and you just hope that nobody scoops you. And so, of course, it was COVID. Everyone was just like hyper-focused on COVID research. And it's like the first day, paper comes out, scoops our top hit. Like, oh, dang it. Next day, another paper comes out. Oh, dang it. I'll just say, while this paper was under review, 50 papers came out, and we were checking off every single one of our hits. And in the supplementary table of this paper now, we have 50 papers while our paper was under review that scooped us. So, well, ours was an algorithm, so we're okay with that. But it was really exciting to see that we weren't just looking at bioinformagic. It was real. The signals we were getting was real. And of course, there's going to be a lot of false positives you expect and so forth. We were just surprised at how few we got, or at least how many of our top hits were not validated. So it was really exciting to see this. We have gone through, anytime you're doing a bioinformatic data package, everyone tries to download it. They can't get to work because packages don't match and everything like that. It's a nightmare. But we went through, we're working with Julio Rodriguez. He's some major director at the EBI. He just recently got promoted to that at the European Bioinformatics Institute. And they have a package called Liana that's focused on cell-cell communication analysis. They've been able to bring in all the different algorithms, all these ones that can only handle 1 or 2 samples. And we were able to incorporate all of those with our tensor cell-to-cell package, so that now you could use all these different approaches that people have to work with larger data sets and so forth. So it's been a fun collaboration that we published this last year. But it's really fun doing these computational studies where you can go through, develop a new algorithm, and kind of see some real cool science come out of it. On that, back to the other side that I mentioned earlier on, where I kind of have this passion project around autism now. This was a collaboration that we started about a little over 10 years ago, when this colleague of mine asked me, he's like, "You're a systems biology, you want to analyze a bunch of data?" I'm like, "Sure, let's do it." And when we got the NIH R01 funded, I was like, "Oh crap, I don't have to do this." And in that process, we were able to generate a very large data set. We were able to get transcriptomics on the blood leukocytes of kids that were very, very young, anywhere from 18 months to 4 years old. That phase when some parents are starting to catch on that maybe their child is a little bit different. And it's really difficult because sometimes people can go years without a diagnosis. My first child was diagnosed at age 11. And as my wife and I, we jokingly, but not jokingly, looked back and said, if we had known earlier, we would have not been so crappy parents. No, we would have been able to meet her needs better. And so now she's a teenager and she's thriving. And in part because we've been able to learn about what are her specific needs. So what we want to do is we want to understand, can we diagnose it a lot earlier? diagnosis predominantly is based off of psychological and behavioral testing. And so it makes it, you have to be, so that's where oftentimes kids start getting diagnosed when they're in school, at school age, because they start to maybe act up or struggle in certain areas, okay? Could we diagnose them at 18 months? Actually, there's new methods that are emerging that are allowing us to do that. And some of the stuff that we're doing is hopefully contributing to that. But so I'm going to give a little bit of information about the diagnostics approaches, but also looking into the molecular bases of dysregulated neurodevelopment and autism. So just to kind of bridge from the tensor cell to cell stuff that I just presented, I'm going to show right here an example where we used it. Now, the autism spectrum is a spectrum. You have a lot of different traits that are common. For example, changes in the rates of epilepsy, feeding difficulties. My kids will only eat specific things. And if I ever change the way I cook something, kills that food and never cook it again. That's why McDonald's is really good for them, because it's always the same. The same age too, but no, but there's sometimes feeding difficulties. You have sleep problems, sometimes social interaction deficits, stereotypies, which is kind of sometimes stuttering or repetitive pattern or repetitive actions and so forth. And now these are all shown right there as kind of like averages, but really you have the whole spectrum. One of my kids is 99.9th percentile in social communication skills. She is amazing. She was a little kid. She would have deep conversations with adults. Yet she can't spell. And then our youngest child really struggles with communications. We ended up being very delayed in her speech. Okay, but both of them are on the spectrum. And so what we do know, however, is there's a lot of similarities in the neurodevelopment. You'll have various levels of AAs of autism severity. Autistic development, oftentimes you have more cells. You have a higher number of proliferating cells early on. And then as they start to differentiate, you get more neurons. Oftentimes in certain brain areas, those neurons might be less developed and less active. And that you also sometimes in some areas of the brain, you get decreased synapse formation and differences in activity when you look at the brain via fMRI. So these are some things that we know from the cellular to the macro scale. What we do know at the molecular level, however, is autism is highly heritable. Its heritability is 83%. That's on par with height. Okay? So when all three of our girls were diagnosed, my wife looked at each other. I was like, "Okay, whose fault is it?" Wonderful kids. They're wonderful kids. And then we both looked at our families, and I realized, oh, my dad was actually pretty different. He always had to wear the same pajamas to work when we worked from home, and my mom could never change the laundry detergent that was used for our family clothes. And he had certain foods that he just would not eat. And so we look back like, okay, he was very much what used to be called Asperger's. Though he passed away several years ago, so we don't know for sure, but my nieces and nephews, I have so many ones. So yes. My wife, on the other hand, she's looked at her family. She's actually seen that there's a lot of evidence of autistic-like behavior among her siblings and possibly her mom. So my wife's also said she's also thinking that she's like, actually, I think I have several of these too. So my wife now and I realize, okay, our kids got won the autism lottery for genes between both of us, and it's brought them wonderful insights into life, and they're doing awesome as their needs are being met. The one thing I bring that to point out, what we know about the genetics, we do know that in general, you get an increased number of copy number variations, loss of function mutations, and even synonymous and missense mutations along autistic people with their compared to their unaffected siblings. And so there's another study where they looked at nearly 5,000 autistic individuals and looked to see what mutations were there. What were they in? The top one is in 24 of the individuals. So there are hundreds of autism-associated genes. And so we have the genetic heterogeneity is huge, yet the phenotypic heterogeneity is not, I would say, is not as big. And that's where you start to think about maybe where do these might converge. That's where you start to have to thinking about these things as a system, as a systems biologist would think about how are they all connected. One thing we do know is that there's really, when you look at the brain development, anywhere from 8 weeks post conception to birth, and then 4 months after the 8 years, there's a data set where they were able to look at autopsies and look at the gene expression of those fetuses all the way to infants and small children, and do gene expression profile across all different areas of their brain. And when you look at it, a lot of the autism risk genes, the mutations that they associate with autism, show their highest expression in the first trimester of fetus development. And then you do have a subset of autism genes that have the highest expression right before birth and the child. So what we do know is that these autism risk genes are more associated with proliferative processes, cell duplication, cell cycle stuff, whereas this group of autism risk genes seem to be associated with more synapse formation. And so we can see that there are these processes going on. And so one thing that collaborator of mine, Gene Yo at UCSD, he went through and he took a number of the what we call syndromic autism genes, where they know that a certain mutation in that gene leads to a certain class, a certain type of autism. And he was able to go through and knock out those genes or introduce the mutations to make in the iPSCs, stem cells, and then culture them into little brain cortical organoids, little mini brains. And we were able to look at those to see how did that affect the development of those brains. And so we did single cell RNA sequencing on them, and you can look at them at different time points. You can see if they did change as the brain cortical organoids matured. You got different cell types, you could see the samples would actually spread out. They weren't overlapped with each other. We'd see different cell types would actually form different clusters and so forth. And this right here is a map of the different mutations. And so you do see certain clusters where different mutations are kind of represented in different regions of different cell types and so forth. But there was kind of overlapping all over. And so what we did is we took our tensor cell-to-cell algorithm on this to see what are the dysregulated signals in neurodevelopment. And so we took these and we had to take the 4-dimensional communication tensors and then bring in a 5th dimension, because we have not only different sender receiver cells, ligand receptor pairs, we have different time points and also different genotypes. Okay, so I'm glad I didn't have to do this on a piece of paper or anything like that, this decomposition, but we were able to use the tensor decomposition algorithm to then get all of this. This is a mess, sorry. But each of these is a different factor. These are the 12 most dominant factors. You can think of these the strongest signals right here. And so right here, we have different genotypes. Here's 4 of the different mutations. These are the different time points. We found that most of the signals, most of the dysregulation we saw was very early stages of brain cortical organoid development. It kind of connects to what we were seeing before. In general, when you look at autism risk genes, they're oftentimes impacting, they have their highest expression during the first trimester of an infant, of a fetal development. And so with that, we're seeing the signal early as on. These are the ligands and receptors that seem to be associated with each of the factors. And then we can look and see the individual cell types, where you have the sender cells here. For example, right here, this factor is predominantly an excitatory neuron effect. And then these are the cells that are receiving the signals on that side. Now, what I want you to get at, it's a lot of data, but what I wanted to see is that we had a few different processes that really popped as being most important. We had cell adhesion molecules that are critical for brain organization, cellular organization of the brain. These are the physical interactions. And we were seeing some changes in those. We saw CD99, that's actually a sex-specific gene, not specific, but it is very differentially expressed between males and females. we actually see that in the autism spectrum, it is diagnosed about 4 to 5 times as frequently in boys than women. And so there does seem to be this difference in response. And then we found a few other pathways. We found several that were associated with PI3K and RAS pathways, and then also Wnt. And these were really interesting to us, in part because we had another study going on that we just completed when we were doing this that overlapped very nicely with this. So I'll talk about that in a moment. And so we were wondering, are these pathways really relevant to autism neurodevelopment? And so this was the other study. We had found those pathways. Do we actually see them? And these are in brain cortical organoids basically derived from iPSCs taken from small children. We looked then at the transcriptomes of their blood leukocytes in the diagnostic space. And so what we want to know is, what were the pathways that were differentially activated or differentially repressed in real children on the autism spectrum? And so we had blood samples from a few hundred kids. We did the transcriptomic analysis. We looked at differentially expressed genes. Right here, we got the differentially expressed genes. And we were wondering, okay, you have these differentially expressed genes. Are they connected in any sort of way? And so we took a number of pathway databases and so forth, and was able to connect them, connect the differentially expressed genes to each other to get a perturbed network. And so what we have here is, this is a differentially expressed gene network for autistic kiddos. And then we had, and to give you a little bit, so that's a big network of genes. We really want to understand, what are the coherent signals going through that? Not just differentially expressed genes, but what are things covariant? The idea here is that if you could have a pathway here, you might have some genes are modestly upregulated, some are modestly downregulated. You don't really think that's going to have a major, that might just be noise. But, and so when you look across the population, if it's just randomly kind of fluctuating, there's not much signal there. But if there's steady increases or decreases, then that might suggest that if you're able to correlate that with some sort of phenotype, then that might suggest that that pathway or that cluster of genes might be driving some sort of signal. And so that's what we did right here. We took our differentially expressed gene network for autism, overlaid the data set on it, and identified specific modules of specific pathways through there that were basically very covariate across subjects. We weren't looking at any phenotype. We're just saying across subjects that these pathways covary. And that gave us a way to basically pull this down to what we call the DEASD network, which is the genes, the pathways within the network that covary when you looked across kiddos on the spectrum. And so what we found here is when we looked at this cluster, right, this is our DEASD network right here. It was mostly just differentially expressed genes because that's what we defined it to be. And then we looked at the autism risk genes. We actually found that there weren't a statistically significant enrichment of autism risk genes in this network. They were all one step away from it. So they were regulators basically affecting that. So we found this, so we took this network that had the differentially expressed genes and the autism risk genes attached to it, and looked to see what sort of pathways were involved in here. And so you saw a number of pathways: mTOR, AKT, PI3K, beta-catenin. We also saw processes like neurogenesis, neuron maturation, and things like that, learning, memory, behavior, things you'd expect to see in autism. And this kind of goes back to that earlier plot, the heat map I showed, where a big chunk of the autism risk genes were associated with synapse formation, so neurogenesis and synapse formation, whereas the others were about we're regulating cell growth and so forth. And that's where we're getting the PI3K, AKT, and regulators, so forth. So we're seeing right here that the growth aspects are getting covered as with the neurogenesis and synapse formation in here. And so right here, now I highlight these because, again, those are the ones we found when we were looking at the single cell data in the brain cortical organoids. Now, so the nice thing about brain cortical organoids is that we identified the ligands and receptors that were driving this, but we also found the cell types that were associated with dysregulated brain development. And so, one thing we were wondering is, could you take these pathways? Are they actually predictive of the autism spectrum? Because yeah, it's nice to know the neurobiology cellular level behind it, but ultimately you want to be able to diagnose it earlier. And so what we found is that we took right here what's called the Autism Diagnostic Observation Schedule. It's one of the major tests that they use to diagnose autism. And it has two components. It's looking at the restricted and repetitive behaviors, and also social and communication behaviors. And it ranks the child in these areas, gives it a score, and above a certain threshold, they're determined to be on the autism spectrum. And so we took these scores and we correlated it with the gene expression in those pathways that I just mentioned, the PI3K or AKT and so forth, and checked to see if we saw in the blood of a child when they were 18 months old, and we knew that they were going to be diagnosed 4 years down the road, how severe was their autism going to be? Could we predict that? And so we took their transcriptomic data from the 18 months, overlaid it on the network, and we found that the score that we got from how dysregulated the gene expression was actually got more and more dysregulated. So the transcriptomes in the white blood cells at age 18 months was going to predict the severity of their autism at age 4. So now you can start to think about, from a simple blood diagnostic, you could catch, so at least the more severe cases of autism based on that. Now we also have our collaborators have developed behavioral tests and so forth that you could aggregate on that. We're doing research right now to see how do those overlap and could we get really strong multimodal diagnostics for autism. So fun research, really cool stuff that would have helped my kids if we'd had it up to 18 years ago, but hopefully we'll be able to help other people. So now the last question is, do any of these findings open up opportunities for therapeutics? And I'm going to go over this pretty quickly. This is some work that's not published yet that we're working on. But again, pulls in the tensor cell stuff and single cell omics to look for therapeutics. And so with this, again, there's lots of different traits here, and lots of strengths, lots of struggles. And so in the speech language delay, I gave that example of my oldest daughter, who is incredibly articulate in speech, but you never have her write anything down for you unless you're using it with Grammarly. And then our other child that is still very much speech delayed. You have these differences here. And then I will say that when I was 20, I was in a part of a service organization where I was actually helping take care of a young boy, 7-year-old. And the whole time I worked with him, I would go visit him every week so the mother could have some time to get some stuff done. And all those months I was working with him, I heard him say one thing, "Huele a limón," or in Spanish, it smells like lemon. And that was what he said when I used my hand sanitizer. And so you have these that are non-speaking where they have fewer than 20 words that they really use. And so with that, there is a really great opportunity here that if we could find some therapeutics to help them to improve their speech, it could be a great benefit for their lives. And so you'll have these kids that sometimes will be considered nonverbal and so forth, as I mentioned. Now, we can't really do therapeutic screens on small children. There's ethics considerations there. And unfortunately, dogs, mice, rats, they don't speak well. But zebra finches do. The interesting thing about zebra finches is that they, like us, they have certain speech development periods that are critical for their speech to solidify. And so when they're young, they'll be listening and they'll gather the songs, and then they'll learn the songs, and eventually there'll be a crystallization of their speech or their singing that will follow the kind of the patterns and so forth that are unique for zebra finches and so forth. And let's see. Now, when you go into the neurobiology, there was an interesting study that when they looked at the brains of zebra finches and non-singing birds, non-songbirds, and then they compared human brains to primates. And we found that there was this region of the brain right here that was pretty similarly located with similar neuron types and so forth that were critical for speech learning, for learning speech and for crystallization of speech and so forth in humans, and then songs in the birds. So you call this area X. In zebra finches, they didn't really actually know what neurons were critical for that, but they knew that roughly where it was at. And so with my collaborators, we looked at some zebra finches, and we looked at the idea of what can we learn about songbird cell-cell communication, and what will that teach us about human communication? And so with the idea that we're looking for some targets. So we did single cell RNA sequencing on that region X in the songbird in birds that had stereotypies. So these stuttering patterns and so forth that you frequently will see in autistic kiddos. We went through, and then we also took it in the young birds versus the old ones. With that point, I'm going to catch that point before how it changes when it crystallizes. When we did the single cell RNA sequencing, we look at the different birds, whether they're non-singer adults or singer adults, and juveniles also. We were able to identify ligand receptor pairs that seemed to be differentially activated among those. We were able to look at the sender cells and the receiver cells. And something that popped up right here we definitely saw a change in certain neuron types that were being dysregulated, and also a decrease in reception of others. So there's a few different right here. Like you see right here, these ones are very much activated and so forth. Now, among these three major factors, we saw a huge separation between the signal among juveniles and adults, suggesting that these might be the critical signals for crystallization. And one thing that we found was that when we looked at the individual ligand receptor pairs, kind of small to see right there, but we found that there were several cases where the cells that were identified basically had this marker for ARPP21. It's actually a marker for the medium spiny neurons. And what was interesting is that in the middle of this gene, there is a microRNA that is in one of the introns. And this jumped out at us, for most people, but it looked for us because this is called microRNA 128. It is something that we'd already seen. When we looked at the transcriptomes of those kids, of their blood, we found that in that dysregulated network I told you earlier about, it was very much enriched in MIR128 targets. There was also a lot of evidence about how this microRNA was important for neurodevelopment, for maturation, migration, and so forth. And as I mentioned, we found in our data. We also had another study where we were looking at fMRI, where we've had, and then correlated that with the gene expression. And we found that the clusters of genes that were correlated with some of the changes in brain activation in these kids actually were also enriched in MIR128. So what we did find was in the birds, going back to the birds, we found that as the birds learned more songs, more motifs, and had more heterogeneous, I would say, song. In other words, more heterogeneous is less controlled, which is actually these two, a lot where the stereotypies come. So this would be more of the dysregulated song. They have higher expressions of MIR128. So they have this signal too. And so what we did is we put in a MIR128 sponge in the brains of those birds to see if we decrease the expression of MIR128, does that change the crystallization, the learning of their singing? And it's always fun to do this when it works. So you have right here, this is kind of a visualization of the song patterns over time and their different frequencies. And so you can listen to some bird songs. These are basically from birds that have stereotypies, a lot of the stuttering patterns and so forth. You have more motifs. Some of them are unclear what they are and so forth. Play. Oh, you can't hear it. Anyways, trust me. I'm seeing really well. So, and so this is pre-treatment. It's you have a lot of unknown motifs and so forth. A lot of stuttering right there. And after we introduced into the birds this sponge, basically knocking down 128 and put them in a cage with a typical singing adult, they were able to relearn, even though it was post-crystallization, they were able to relearn the proper song. So it was able to restore proper singing. And so we're excited, but I mean, how many parents would let you inject a sponge into their child's brain? I would probably wouldn't want to do that with my kids. So, and so there's got to be something better. And so we'll just show right here. This is the data showing where we go from pre-treatment. This is the typically reared birds. That's scramble control. Right here, these are the birds with the stereotypies. And then after we added the sponge, it brought it up to match the typically reared ones. Okay, and that sponge right here, this this control right here, that was from the dysregulated birds. So the sponge the control did not do it all. So we're excited about that. That's where we came across a small molecule. It's a natural product. It's in an herb. Very, very, very low concentration. But we identified it, and we found that with that, if we added that to cell culture, we could reduce the expression of miR-128. So like, okay, this is cool. Let's try this on the birds. And it did the same as a sponge. So now this is a small molecule in an herb that's considered as safe for consumption, that if we get it in its purified form, we very well could be treating various stereotypies. Is it going to work for all autistic kiddos? Probably not. Will it work for some? I think it might. So we're in the process right now of trying to set up the clinical trials to be able to see if this could be a new therapeutic. So hopefully I'll be able to tell you some good news on that. If not, it's still an interesting story. So, but in summary, so cell-cell communication, I hope you convince you that cell-cell communication is really important. We need to be able to disentangle these signals between all the different cell types. And as complex as the cacophony that you'd have inside of a tissue might be, we can use single-cell omics and spatial omics. So we didn't show any of that, but you can use spatial omics also to be able to start to disentangle these different conversations. And that can guide further studies into understanding how the cells are communicating with each other and making decisions as through development or in response to disease and so forth. And that we can find these cell-cell interactions from single-cell omics. And our approach is one. There's a number of methods out there. We did a review last year, and there are already a couple hundred. I get asked to review these new algorithms like on a weekly basis. So there's a lot of them out there, and it's continuing. We're starting to pull together a consortium of experts in this space to kind of build up interest around the human cell communicome, however you want to say it. So to really map out all the different interactions between cell types and when they're important. So we did apply this to brain cortical organoids that are developing, and we've been able to identify patterns not only from the transcriptome. This was from the kiddos, from their blood transcriptomes. We found the highest expression of various signals early of autism associated signals early in the first trimester, and that those were predictive of severity. But then also we can now go in and identify ligand receptor pairs to suggest potential therapeutic targets, at least relevant to songbird stereotypies, and very likely because of the similarities between learned vocalization in birds and humans. We are now looking into, and we do have data on humans, but we're moving forward to hopefully be able to look at this as a potential therapeutic. Yeah. So this work was led in large part, this was in a collaboration with Stephanie White's lab at UCLA on the songbird stuff, and some of the stem cell stuff was done with Jean Yo and Eric Crochet and Karen Pierce. Intercellular cell was done with Rob Knight. He does a lot of microbiome. Stole one of his algorithm ideas and collaborated with his students. We collaborated on that. But the songbird stuff was special. It was led by Caitlin Ahmad, outstanding postdoc in my lab, and also a few alumni from my lab, a few bioinformatics PhD students. So, so that's that. I will say they wanted me to kind of give a few pointers on how I mentor bioinformatics students. And there's a lot of cool stuff to learn here at PhD. Your PI might just be asking for papers. Hopefully you're getting more than that from your PhD. And so the research is obvious. I mean, there's going to be, in my lab, we do a lot of systems biology, network analysis. We do biomanufacturing, we do cell culturing and so forth, single cell omics and neural networks, stuff like that. So lots of fun stuff in there. Now we're getting some automation too. And that you're going to get that during your PhDs, but there's a lot more. And so in teaching, you do have your subject-specific curriculums that I've taught, systems biology, bioprocess engineering, stuff like that. But another thing that I really pushed a lot back at UCSD is we taught a class on communication for our bioinformatics students to help them to be able to pick good projects, but also be able to communicate them well. Because half of good science is communication, how you're able to project it, how you're able to share it with those that are not in your field is absolutely critical. And so that was a book we would use that I wish I'd had when I was a student. I didn't find it until I was brand new faculty. But that's another thing. Now, another big thing I always pushed in my lab was internships and industry interactions, because a lot of my students would go to industry. I had some that wanted to go academia, but a lot of them would go to industry. And so we would pair them up, get them internships throughout their PhD, either at various companies or sent some to Portugal or sending her to Portugal, hopefully soon. And then Denmark sent several students out there. So it gave them opportunities to learn something new, to get out of their comfort zone, and especially if going to companies, to get a sense of what it's like, what's the culture. Science is approached differently. There's some great skills you can gather from there. Also entrepreneurship, we've spun out a couple companies. This is a bioinformatics student of mine that had this crazy idea of being able to predict light structures from protein structure. Spun out a company and recently got an award as a finalist at the Recode Edge competition. And so he is super busy right now and trying to keep me busy too, as we are scaling that up. And then we have some help with another company like in sequencing. And then outreach. We're not just our own little bubble right here. There's a lot of scientists globally. You want to be able to reach out to them. Eric Armigall, he was the one that developed tensor cell to cell. We got into this program where we were able to help organize and present this outreach to the Human Cell Atlas Latin America consortium, all about cell-cell communication. We did that, and then advocacy also. This is Ben Kelman. He was a grad student who was concerned about the direction of science. And so he basically told everyone, give me a bunch of money, pledge me for how far I can bike, and I'll bike all the way from San Diego to San Francisco. And so he put together this thing. He was able to raise a large amount of money that he was able to then donate for science advocacy. And the AAAS wrote an article about him. I biked with them for like few miles. It was really fun. And these sort of things you can think about. How do you communicate science better? How do you connect with those that think differently than us? It's so critical. So anyways, that's my little spiel. Thanks. All right. Sure.
As someone who's on spectrum, good care and which you handled this whole talk. But I actually have a few questions. Were you on NPR or one of your collaborators on NPR like a week or a month ago?
On NPI?
NPR.
Oh, NPR. I wasn't.
Okay.
So I thought I don't do public speaking.
Okay. But anyway, I was actually, and my second question is how many birds do you have to go through to find phenotypic stuttering birds, I guess?
This was a case where there's certain treatments that you can do. You can raise them in certain areas, or you can do certain treatments. There are certain
Yeah.
treatments they know that alter brain
Okay.
development,
So you do so.
such as valporic acid and so forth, that actually hits those pathways, dysregulates those pathways that we identified. So yeah, there's ways, that's ways you can do it. Yeah. Other questions? Yeah.
You mentioned about ASD doing same things. I was wondering, can OCT be linked with ASD, and how do they overlap with each other?
So genetically, if I remember correctly, there are comorbidities there. There's some overlap. A lot of these neurodivergent conditions, autism, ADHD, OCD, several come in. They overlap genetically. That was something that blew my mind when I was a grad student when they did the first GWAS studies and they showed that. And that's why you look at my family. I'm not on the spectrum. I have ADHD though, and we see that kind of sprinkled throughout our families. You'll have a lot of enrichment of various people with different conditions. With that, some of these techniques can be and are being used for studying other conditions such as OCD and so forth. So there's a lot of opportunity in this space. It's amazing that what we're able to start to tease apart. Now it's still so complex, but more data, sure.
Yeah, so you mentioned at the beginning that you have a data matrix and then you reduce that data matrix with just ligands that, bind to certain receptors.
Yes.
And then you talk about your natural product at the end. I was wondering if you could, how do you find the ligands that bind to receptors to use correlations to find novel interactions?
There's a lot of different techniques you could use for that. And one of them is, like what you said, you don't necessarily reduce your whole data set. Sometimes you look at gene clusters and say, well, these genes I know are associated with this condition. So we could actually go back and look at WGCNA, gene co-expression analysis, and identify, okay, we know the autism risk genes are these ones. And then we find these ligand receptor pairs. Well, what about these other ones what about these other genes that we know are secreted, but aren't really associated, aren't our ligand receptor pair database? You could include those in the data sets as new ones. And then you can also go back to your analysis and say, well, what other genes had the same pattern that we'd originally filtered out? There's a few different ways you can go about doing that, but that's so important because we don't know everything about cell-cell communication. So, yeah.
Nice talk. Good. I noticed both insulin pathway and mTOR being dysregulated in autistic people. And is there a different incidence of diabetes and autistic?
There's different metabolic enrichment of metabolic disorders in the spectrum where they're increased. Yeah. And it's oftentimes thought, well, maybe it's just because of the altered eating patterns. But I think it's from our data, it's suggesting that it really is. There's a dysregulation of some of these. About half of the autism risk genes are broadly expressed across all tissues, and so that's where you're able to get signals of a large number we're able to predict autism from the blood, for example. It's because these are dysregulated, so they are affecting metabolism. They're affecting a few other conditions. So there's a lot of comorbidities. So, yeah. Other questions? Sure.
Quick one from Zoom. How large are the blood samples for the diagnostic test for the children?
The volume,
Yeah.
Here's like a milliliter. It's really small. You use as much as you need for transcriptomics. So it's pretty small. And but then as far as the end goes, we did that first study on 200 subjects. We have several. We have a few thousand now. So.
All right. All right. Last question, and then in the interest of time, we're going to move on. Yep.
I'm curious about the stereotypic birds. Before the treatment, some of them, even the more normal birds, they do have some unidentified patterns, but after treatment, they all look the same. Do you worry about the treatment will kill some of the creativity of their brain?
That's actually a good thing to consider. That's where the whole concept of treatment in autism and neurodiversity is kind of a touchy subject because these are members of our community that bring in unique aspects that are incredibly valuable. A lot of science, there's an enrichment of scientists that are on the spectrum and have ADHD, so forth. And you may not want to stifle that. And so that's where you have to kind of think about this carefully. You have to proceed with these things carefully. In this case, the birds were able to learn the patterns better. And whether there are positive or negatives, that's something that you have to kind of think about on an individual basis. I will say there are certain struggles my kids have gone through that I am grateful for the therapeutics that helped them along the way, because it brought them to the part where they cannot function to where they can. And so it's a delicate topic, but you bring up a very important point.
All right, if there are more questions, you feel free to stay and ask afterward. Otherwise, thank you.
Hey, we're in 2017.